welcome to Particle Metrix talk about insights into multifluorescent nanotracking analysis based extracellular vesicle characterization. My name is Christina Klaasen and I'm working as a sales manager for Particle Metrix. I will start with a short introduction about nanotracking analysis in general to bring everyone on the same level. As you might know, with a nanotracking analysis, we track the movement of small particles, like for example extracellular vesicles or exosomes. This movement is called Brownian motion and it is dependent on the particle size. That means by analysis of this movement, we get to know the size of each single particle, we get to know the count or the concentration of our particles in our liquid, and one special feature of our NTA system named ZetaView is the analysis of the Zeta Potential, which is describing the surface charge of our particles. I'm pretty sure you are familiar with this kind of size histograms where we plot the size on the x-axis against the concentration on the y-axis. Additionally, we have the possibility to show the data in a dot plot, where we plot two different parameters against each other, like here the size on the x-axis and the mean intensity on the y-axis. But there are a lot of different parameter options available, like for example the Zeta Potential, the particle area or shape. And these dot plots give us the possibility to divide between different subpopulations pretty easy. Now, I gave you a brief overview about NTA, but the title of my talk says fluorescent NTA. Why do we need fluorescence at all when we can analyze all these parameters in scatter mode without fluorescence? The answer is easy. In an NTA device, our particles look like this. They are bright stars in front of a black sky. But how can we be sure that we are really looking on our exosomes? Couldn't this particle be a nanobubble or a protein aggregate, an insoluble salt precipitate? All of them have roughly the same size and the same scattering abilities, meaning that they look the same in our NTA. But this is exactly the way conventional NTA works. All particles in the sample are illuminated by a laser. All particles scatter light, which is detected by the camera and analyzed. So all particles, no matter what they consist of, are part of the analysis. The solution for this problem is absolutely easy. We simply put an emission filter in front of the camera, so the laser light is blocked and only the longer wavelength fluorescent light is analyzed. So we can forget about impurities and focus only on our particles of interest. That sounds perfect, but there is a disadvantage. If we want to work with fluorescence, we need some kind of fluorescent staining. The easiest staining you can do is a membrane staining. There are thousands of lipid dyes available, which integrate into the membrane and stain thereby any particle with a lipid membrane. These dyes have a lot of advantages. They are available for any laser color, they are bright, cheap and easy to use. Here I show you an example of lyophilized exosome standards from HCT 116 cells, which have been stained with cell mask orange. In blue, the size distribution of the scattered channel is depicted, and in orange, the result of the fluorescent channel. As you can see, we find 85% of all particles from the scatter mode in the fluorescent mode as well. This is a good result, showing a very pure sample, but it means that 15% of all particles have been rubbish. And if you have a look on the median size, you find it influenced by this 15%. And as I mentioned, this is a very pure exosome standard sample. You can imagine that the influence will be much higher in any other kind of sample. So we have a very good tool to improve our results with a membrane staining. But you might argue now that there can be other kind of particles in our samples which have a membrane but are no exosomes. And of course you are right. So if you want to work with more specificity, you have to work with antibodies. For exosome identification, we use a triple staining with the three exosome markers CD9, CD63 and CD81, all labeled with the same dye, in this case Alexa Fluor 488. We used the same exosome standard as I showed you before. In green, you see the result of the fluorescent channel, and in blue, you see the result of the scatter. We performed this experiment 4 times, and if we have a closer look on the concentration, you find roughly 65% of all particles back in the fluorescent channel. If we remember our finding from the last slide that 15% of all particles didn't have a membrane, we can assume that 30% of the membranous particles present in this standard are no exosomes. If we have a look on the median size, you can see that the influence of this 45% of particles which are not of our interest have a great influence on the result. Summing up this slide, I hope that you agree that if you want to have reliable NTA results, you should use the fluorescent channel and not the scattered channel alone. Of course, you can use other antibodies than we did. The antibody market is huge. There are a lot of different antibodies with different dyes available for all laser colors that we offer. Some are more suitable for fluorescent NTA than others, so please get in touch with us in case you need assistance. We have optimized our ZetaView for fluorescence during the last few years. Since last year, we have, for example, a software feature which is called low bleach function, and this prevents sample bleaching. We developed the quad system, which can analyze 5 different channels. Since we have 4 lasers integrated, a violet one, a blue one, a green one, and a red one. So we can analyze the scatter in 4 different fluorescent channels, all with the same sample. That helps to characterize your particles very properly while saving sample volume. So we can do multi-fluorescence analysis. As you might remember, this was part of the title of this talk. So the question is, do I need the quad to do multifluorescent NTA? Good news, the answer is no. Even if you have only 1 laser, you can do multifluorescence. But how does this work? We developed a new feature which is called multifluorescence filter. As you know, fluorescent dyes can have different Stokes shifts. In this example, we see Alexa Fluor 488 and PerCP. Both of them can be excited by the blue 488 laser. The Alexa Fluor 488 has a Stokes shift and emits light in the green wavelength area. The PerCP has a long Stokes shift and emits light in the red area. If you're using the conventional filter, you're using a long pass filter. So you can analyze both of them in the fluorescent channel, but you will not be able to find out which proportion of the signal is coming from which dye. But with the new multi-filter feature, you can use the blue laser, but select a filter which blocks the green light, so you can detect only the PerCP signal. For detection of the Alexa Fluor 488 signal, you can select a bandpass filter, so you can do multifluorescent NTA with only 1 laser. We can update all ZetaView 120 machines with this feature. If you're interested, please get in touch. Now I'm coming to my last topic, which is how to do fluorescent NTA in general. I'm sure most of you are doing other kind of fluorescence analysis, like flow cytometry or fluorescence microscopy. So you might have already protocols for staining. But these protocols are often not usable for F-NTA. Why not? We have to understand the challenges to overcome them. Challenge number 1, we are working with fluorescence and with stain with a high amount of dye. So it would be good to wash all the unbound dye away. But unfortunately, we are working with very small particles. Washing is a problem. It would cost us a lot of time, and we would for sure lose a portion of our signals of interest. Since we cannot wash, we are dealing with a lot of unbound dye, so the signal-to-noise ratio is a challenge here. Challenge 2, antigens are rare on exosome surface. On a cell you can have tons of antigens, but exosomes are a lot smaller, so you have only some antigens on an exosome surface, so only a limited number of antibodies can bind. Limited numbers of antibodies means limited number of fluorophores, so we can only get a weak signal. So we have to be very sensitive to detect this signal. And we are dealing with bleaching. Why is bleaching such a problem? In flow cytometry you don't talk about bleaching. Why do we do? In flow cytometry your particles run through the laser beam and are instantly analyzed. But we do a tracking analysis, which means we have to follow the movement of the particles at least for half a second. So we have to care about bleaching. How can we deal with these challenges? What are the solutions here? we decided to work with no-wash protocols to save time in vesicles. But if we do so, we have to work with a very low antibody concentration. In most cases, we need a pre-dilution of the antibody. You should titrate your antibody to find the ideal signal-to-noise ratio and select your fluorophores for F-NTA with care. Use bright fluorophores, since only a few of them can bind to one exosome due to the limited number of antigens. And use stable fluorophores, since we are tracking our particles to calculate the size. And last but not least, use the low bleach function. In case you are using an old software version without this function, please get in touch. We can update your instrument. We can also help you with protocols, for example, for the triple staining I was talking about. Now I'm at the end of my talk. Thank you very much for your attention and for your interest in fluorescent nanotracking analysis. If you have any kind of question, I would be happy if you get in touch with us. I hope that we can meet each other in person next time. So stay safe and enjoy the online workshop. Thank you and goodbye.